Hello, Science 10s. This is the first lesson of biology. So, we will get going with it. So, again, we're going to get through most of biology, but just due to time constraints, we might not actually get through everything, but it's probably the stuff at the end that will get smushed up a little bit, so the stuff related to plant stuff. So I know quite a few of you guys are interested in taking Bio 20, so a lot of these are very foundational for what you'll need in Bio 20. Bio 20 is a very heavy course for information. It's an awesome course, but there is a lot of information, and some of this cellular structure stuff is important for your understanding in Bio 20. So I'll gloss over a couple of things from the notes. I didn't modify them or anything, so if I say we're skipping over something, you'll just have to be okay with it, or we'll zoom through it pretty quick. So we'll do B1 here, so Bio 1, student notes. So what is biology? So I'm a biological sciences major, so bio is probably my number one. I like chemistry too, and then other things kind of fall beside both of those. So biology is the science or study of living things. So bio, the word bio from Latin means life or living, and the word ology again means study of. So I actually took a really cool course in university. It was called Greek and Roman Studies, and it was actually a medical terminology. So breaking down all of these words into their Latin or Greek derivations and understanding what it actually means. So ology always means the study of, so something like seismology. So that's S-E-I-S, seismology, so is the study of seismic activity, so looking at plate tectonics and movements and stuff like that. Oncology, I'm just trying to come up with ologies, so oncology is actually looking at the study of cancer. Um, my favorite one, I don't know why I remember this, so gymnast, so gym means naked, and nast actually is related to athlete. So a gymnast is a naked athlete. I don't know why I remember that breakdown, but things you remember from 15 years ago. Yes, I am that old, that I was in university 15 years ago. So scientists have agreed that all living things consist of, let's try to use the right words, consist of 5 characteristics. So living things, they need energy, they produce waste, and so they need a way to get rid of that waste. They respond and adapt to their environment. They reproduce. And so that's not necessarily sexual reproduction, but asexual reproduction, so producing new cells. Um, so in Bio 30 and Science 30, you learn about asexual reproduction in terms of mitosis. Um, they also have the ability, can grow. So organization of multicellular organisms, so anything that is multicellular, so plants, humans, animals, etc. The smallest common unit found in living things is the cell, or the smallest unit of what we will call living is cell. There are parts that make that up, so there's just like there's subatomic particles, so something smaller than an atom, the parts of the atom. Um, there are parts that make up a cell, but on their own, they're not considered to be living. Cells actually work together to form what we call tissues. So something like um, particular tissues, the epithelial cells of your digestive tract. So that's a particular type of tissue that's within there. Those work together. Tissues actually work together to form organs. So those would work together with transporting cells across your digestive tract, so form organs, whether it's your stomach or your large intestine or whatever it would be. Organs actually work together to form organ systems. So based on the example that I was looking at there, your digestive tract, that's an organ system. So they're all for a common function of acquiring nutrients from food, and they work together, but different things have different responsibilities, right? Your stomach is responsible for starting to break down things through mechanical and physical means. Different parts of your intestine absorb different types of nutrients, etc., etc. So types of cells. So this, you may have seen some of this stuff before. So something called prokaryotic cells. So these are like bacteria. So prokaryotic means before, holy heck, there are lots of mistakes. I don't have a pen to change it. Before the nucleus. Sorry, I'm just searching for a pen to correct my notes so I fix the notes package. So prokaryotic means before the nucleus. These tend to be fairly simple cells. So they do not have a membrane-bound nucleus. So what's considered not a true nucleus. Nucleus, again, we haven't talked about it in Science 10. We'll talk about organelles in the next lesson. If you took Science 14, you would have seen organelles there. And so the nucleus is kind of the thing that directs the activities within the cell. It's like the brain of the cell. Prokaryotic cells have DNA found in a plasmid form. So their DNA is actually just kind of a ring of DNA because they don't necessarily need that much DNA. They don't have a lot of functions. They're not doing a whole bunch of things. So DNAs are found in plasmid forms, which is actually important in terms of some of the genetic engineering. We can feed some DNA into that bacterial plasmid and actually cause that bacteria to produce things that we want, like insulin can be produced by bacteria if we insert the gene for it within there. Sometimes they can have cell walls. And so these types of bacteria or these types of prokaryotic cells can have kind of three main shapes. There's definitely outsiders on everything. So sometimes they're very spherical or they look like little balls, and those are called coccus. You can have bacillus. So those are kind of a rod-shaped form, or spirella or spiral type where they just look kind of wiggly like that. Bacteria, for the most part, will reproduce asexually, meaning that they simply divide and create an exact copy of themselves. Sometimes bacteria can share genetic information with one another, and that's kind of a weird form of sexual reproduction where they actually insert some of their genetic information into something else, and it's kind of a weird form of sexual reproduction. So that's prokaryotic cells, really basic. Again, if you're taking Bio 20, you're going to look more into some of these things, but this is just superficial. The next type of cell that we'll talk about is eukaryotic. Me, no, eukaryotic. And so the word eu, spelled EU, actually means true. So that is, it's saying it's a true nucleus cell. So they're more complex than the prokaryotic. They have a true nucleus that is membrane bound, meaning that it just has a membrane around it. And DNA are organized into chromosomes. So they're no longer that ring form, but they're into separate chromosomes, which is important for cellular division and reproduction. So two major types of eukaryotic cells exist: animal cells and plant cells. And so we'll look more at both of these, the organelles within them, and what the roles of each of those organelles are. So animal cells can be lots of different shapes and sizes. They contain mitochondria, which we'll look at the function of mitochondria and what it looks like. Mitochondria is important for cellular respiration, and what that is just obtaining energy from glucose. And they do not have cell walls. Plant cells do have a cell wall. They also contain mitochondria for cellular respiration. So plants do cellular respiration, but they also do this fancy thing that we all wish we could do, which is photosynthesis. So they have the ability to capture energy from the sun and convert it into glucose, also taking in carbon dioxide and water, and having another product be oxygen as well. So we'll look more at plant and animal cells. Viruses and prions. Again, this is not super important. I probably would glaze over this really quick, but people kind of like this type of thing. So what is a virus? A virus is a non-cellular structure made up of a stretch of genetic material enclosed in a protein coat. And what a virus can do is it can trick our cells and allow it to enter. Once it's inside, a virus will take over the cell's reproductive machinery. This causes the cell to continue to produce more viruses. So viruses have a lot of different mechanisms of how they accomplish these things and what they actually do within your body. Taking these types of courses is incredible, but also incredibly complex, as we're seeing with information about viruses these days. And I definitely don't have any sort of depth of understanding of the COVID-19 virus and kind of its mechanism and how it affects the body, but viruses are very interesting, but also can be a lot to handle. So some of the viruses, again, I could change the notes for COVID and learn a little bit more about this, but some other viruses that we have information about, a lot of information about now. So HIV versus AIDS. So HIV is the human immunodeficiency virus. And what that virus actually does is it attacks the immune system. So that virus specifically is looking at the immune system and finding a way to prevent it from functioning normally, because if your immune system isn't functioning normally, then you can't fight off things within the body. So this causes the body to struggle at fighting off diseases. Or infections. So HIV infects and destroys certain white blood cells. So they target particular white blood cells. And if too many of these white blood cells are actually targeted and destroyed, the body can no longer defend itself against infection. So when you start to look at immune response and that sort of thing, if you've done Science 24, you would have looked at it in there, but also in Bio 20 and in Science 30, we look at immunity. White blood cells are really important for the immune response. So for informing the body that there is a pathogen or an invader or something that is not supposed to be within the body. So if there are not those white blood cells, then anything can get into your body and you won't have the immune system to back a response. So that's actually the problem with HIV, is now you can't fight off a common cold. You can't fight off just a bacterial infection or anything like that because your immune system is so depleted. So HIV versus AIDS. So sometimes these are used interchangeably, and they definitely should not be. So AIDS, so that's the acquired immunodeficiency syndrome, is where people who have had HIV for a long amount of time, feel like I'm missing kind of a sentence there. So people who have had HIV for a very long time and the white blood cell count is extremely low. So it's reached a point where basically you can't fight anything off. So now you are immunodeficient. I keep saying you just as a general term. An individual is now immunodeficient and they don't have the ability to fight off infection. So they're now, I think I changed it in the notes because I saw this one, susceptible to infections and even things like cancers that rarely occur in healthy people. So these can be quite deadly. So HIV is the virus, AIDS is the symptom that actually comes once somebody's been infected for usually quite a long period of time. So HIV and AIDS facts. So having HIV does not necessarily mean that you have AIDS. So this number is totally up for debate, but HIV takes about 10 to 12 years to progress from HIV to AIDS. And it absolutely can be held at the point where somebody only is infected with HIV and does not necessarily end up being classified as having AIDS. So the example that is used quite often is Magic Johnson, so a basketball player. He was diagnosed, I think, in the 80s with HIV, and he actually, I believe, hopefully I'm not remembering this wrong, has never been diagnosed with AIDS. So he's been on antivirals. I want to say the word retro, and I don't know what word I'm trying to say, but he has been taking what's sometimes called is the HIV cocktail. So it's a series of medication that has actually kept his immune system working at a reasonable level that he has never progressed to the point of having AIDS. So many people are able to live a long life, an active life, with the help of medicine to build the immune system back up and keep it at a point where it's not being constantly attacked by the virus. So HIV doesn't survive well outside the body, so that's why you can't get it from kissing or sharing a glass of water with a person who is infected. So it's transferred significantly more through blood-to-blood contact is the big thing. So it's not surfaces, it's blood-to-blood contact. So prions, so a prion is a subcellular structure that can reproduce in living tissues or cells. It's composed of a protein that converts from its normal form into a harmful particle. The most common prion that you may be aware of is bovine spongiform encephalopathy, which is mad cow disease. So through eating food that's infected or different things that are infected with this prion, cows have a pretty tough time. So human prion diseases include what's called Creutzfeldt-Jakob disease. I think it's an E first. And something called Curu. And so again, we're rushing through a little bit of this stuff. I like to usually show some videos related to viruses and prions and stuff like that, but we don't have too much time. So all known prions affect the structure of the brain or other neural tissue. Illness is progressive and always deadly. So prions are not good. So this is actually, we won't talk about that. So viruses and prions do not have any independent life functions and therefore are not considered to be a functional unit of life because again, they're not cellular. So they're subcellular or they're like portions of cells, but they are not cellular. So lots of scientific research has been done to come up with the characteristics of living things as well as the development of what a cell even is. The next section will look at the history of some scientists who contributed to the development of cell theory, as well as the biggest breakthrough, essentially, in biology history is the microscope, so that we can see things that are super, super tiny. So I think I deleted this out of your notes. I don't think it was in there. Usually I have you guys write down kind of the whole timeline for some of the major events in terms of cell theory and into microscope theory. So this is looking at before Common Era and Common Era, none of this before Christ, after death stuff, so before Common Era and Common Era. So in 384 to 322 BC, Aristotle actually proposed something called spontaneous generation, or what was also known as abiogenesis. And so this proposal was related to organisms arising from non-living matter. And so again, I'm not going to go through all of this. Usually this is kind of an entire day's lesson looking at all of these, but essentially it was you can leave something that's non-living and it can turn into something living. So organisms arise from non-living matter. We know now that that's not true. So good try, Aristotle, but not today. So Hans and Zacharias Janssen, so in the early 1600s, they actually are credited with kind of inventing the microscope. So he used a two-lens system with a magnification of 20 times. So that's the first compound microscope. So before people had types of magnifying lenses or a magnifying glass, but this is using a two-lens system in order to magnify things even further. So Francisco Redi in the late 1600s was the first person to kind of start saying, yo, Aristotle, like 2,000 years ago, you said spontaneous generation was a thing. And he was starting to prove that it's not actually true. So he concluded that when you left gross meat out on the counter and all of a sudden 2 days later there were flies growing on, or there was maggots growing on it, that dead pile of meat did not actually turn into something that was live. What actually happened was flies were laying eggs on the surface of the meat, and that's why there were maggots there later. So Anton van Leeuwenhoek, ooh, that's a cool year. I assume that this would be, let's take out the 9. So 1723, not 17,000. So he used a simple lens microscope with about a magnification of about 250 to view single cells. So he was kind of the first one to view cells and get a little bit of information from it. Robert Hooke, same time-ish, looked at cork from the bark of an oak tree under some type of compound light microscope, magnification about 30, and he actually started the word cells. So animalcules is what Anton van Leeuwenhoek called it. Robert Hooke actually called them cells because they looked like little boxes, so we called them cells. Schleiden Schwann in the 1800s started to look. So you've got a couple of years between these guys. They got more information about cells. Oops, missing the edge of it. And so they started to apply it that animals were made up of cells. And then we get into cell theory. So information that cells come from more cells. So all cells arise from pre-existing cells. And Louis Pasteur did a nice job of creating an actual experiment that's considered a little more valid today. So having a control group, manipulated variable, and responding variable to really and truly disprove spontaneous generation. And so Pasteur, you've probably heard the word pasteurization, right? Like milk is pasteurized. Pasteur was responsible for a lot of that stuff. Milk is pasteurized so that there's not bacteria and stuff growing in it. This is based on his information and his understandings. This is me now freaking out that maybe I didn't press record. Did I? Oh, I did. I'm so good. So don't worry too much about the scientists, but know that it's been a long time to get to where we are today, and you got to make mistakes to get all of the information. So all of the above scientists helped contribute to the development of cell theory. So cell theory, that we're going to talk about 3 parts of it. So all cells come from other cells. All organisms are made up of cells. And the last one we've already kind of talked about. So cells are the smallest functional unit of life. So that's our 3 parts to cell theory. So what about the microscope? So again, bio, usually we spend a lot of time doing microscope labs and looking at stuff under the microscope, but not today. Oh, actually, here, I'm going to pause. Uh-oh, you won't let me. Oh no, what's going on? You're gonna be mad if I screwed stuff up there. I don't know what I did to the screen here. Let's erase. Sorry, I was just trying to get another window open. But let's do this first. So some of the microscope stuff. So types of microscopes. So you can have something just called a simple microscope. And this would be more like a magnifying glass. So you have the ability to magnify 5 to 15 times a regular amount. And so that just gives you an enlarged virtual image of the specimen. Um, we also at school have something called dissecting scopes, which are basically like a magnifying lens. And that just allows, if you're like looking at a human specimen, not human, a fresh specimen. So when you're looking at a heart or an eyeball or whatever else you're going to dissect in your future, if you want to, you can look at it and just get a little bit closer of a look without having to hold something like a magnifying lens. So the ones that we usually use are compound microscopes. And so those are made of 2 or more lenses, usually uses light source. So that's the ones that we have in the classroom. So ours magnify from about, the lowest magnification is 40, the highest magnification I believe is 400 on the ones we have. Some schools, depending on what you have, sometimes you'll have ones that magnify up to 1,000 if they have a 100 lens on them. I don't believe ours do. So other types, so we have something called transmission electron microscope. And so that allows electrons to be transmitted through to produce a 2D image. This is, you gotta have stuff that's sliced very, very thin. But basically what you're doing is you're sending electrons through something, and the way it reflects off is what gives you information. But you can see your magnification is real big. But the coolest one are scanning electron microscopes. They allow electrons to be swept over to produce a 3D image. Specimens don't have to be cut very thin necessarily, but it gets a 3D image of the surface of the specimen. And so I wanted to look up SEM ones. That's what I was doing there. So scanning electron microscope stuff. I don't know what that one is, but so this, they added color to this. So this is a SEM image of pollen from a variety of plants. So this is pollen, the stuff that causes allergies, and the trees are getting rid of, some from sunflower, primrose, and lily. So this is pollen put through a scanning electron microscope. So this is the epithelia of your trachea, so kind of within your throat, and it shows ciliated cells. So these are kind of the hairs on the lining of your throat. That's gross and awesome. You ready for gross? The head and mouth of a leaf beetle. So that's a beetle, tiny little beetle. This is the eye of the fruit fly. So, you know, those little tiny flies when you haven't changed your compost container in the house for a while and you get fruit flies. This is what their eye looks like up close. Super cool. Yellow mite, gross. A bed bug, ugh. This is the surface of a kidney stone. zoomed in. This must be from the fleas of fruit flies. I didn't look through all of these, so hopefully there's not anything too weird. This one? Oh, this is a white blood cell eating a staph. It's like a particular type of bacteria. That's a cell dying. Looks like it. So some algae. Our tardigrade. Oh, I love water bears. They're so weird. And that's about it. But you can look up really cool SEM images. And then there are more complex ones that allow you to see even greater. So scanning tunneling microscopes, I think there's maybe only one of those in the world. They're super expensive, but you can see an incredible amount of information. And so I think in your notes I have the paramecium. So we have paramecium slides at school and you can look at them, and that's under a 400 times magnification. So on our highest magnification. And then this is a scanning electron microscope of an ant. So you get kind of that cool 3D image of it. So that is it for the first one. The next lesson, we're going to look at those organelles within cells and what their roles are. So that's it. I'm just going to pause again because there might be a worksheet. I can't remember. So if I don't come back, there's not a worksheet.